This video is going to have a look at biotechnology. I'm going to focus on what biotechnology is and how it's used on a large scale. So the first thing you need to know then is what biotechnology means. And nice and simply, it's using living organisms to produce useful products or to provide people with services. So what we need to look at is how these microorganisms, which are useful, are grown, how they're made. And it's a process you should be familiar with called fermentation. So first things first, they will take the food, which is provided in a liquid culture medium, and they'll put it into the fermenter. This food will be carbohydrates, which use sugars, which is the energy source, nitrates, which can be used to make protein, vitamins and minerals. All of these added together make a liquid culture medium. So the next thing they do is they'll pump the microorganisms in that they want to reproduce. Step number 3 then is to pump in lots of air. This gives the microorganisms lots of oxygen so they can respire aerobically. Step number 4 is to make sure the temperature is correct at around 37 degrees, which is the best temperature for microorganisms to reproduce. So they'll use something called a water jacket and a thermometer. The thermometer will keep an eye on the temperature, then the water jacket can either be made cold or hot depending on what the medium needs. The next step is to make sure that the pH is correct. Make sure that the microorganisms can thrive, and they'll use pH monitors to make sure that this is correct. They also need to make sure that the conditions are sterile, which means it's aseptic, that prevents contamination from other microorganisms. And last but not least, they'll use a motorized stirrer, and that will keep the whole culture well mixed so all microorganisms get the nutrients and enough oxygen. So what you do need to know now then is how you can investigate the effect of these different factors on yeast growth and on microgrowth in general. So in the example of yeast, which respires anaerobically, it turns sugar into carbon dioxide and alcohol, and you can do an experiment to measure the rate of CO2 production by yeast under different conditions. So the first thing you do is you boil some water in a test tube. That gets rid of any dissolved oxygen, so it's only anaerobic respiration. You then add in your yeast and some sugar, and then add a layer of paraffin oil that stops any more oxygen from getting in. Then you get a delivery tube and connect it up to lime water, which will obviously go cloudy when carbon dioxide is present. You can then vary the temperature, the pH or the concentration of sugar and use this to count the bubbles produced to check the rate of production. So the optimum temperature you'll find is around 37 degrees. Any higher than that, then it will start to kill off the yeast, and any lower than that will be too slow. Varying the pH, you'll find out that it's about 4.5, so slightly acidic is the best conditions. And then in sugar concentration terms, the more sugar, the faster the yeast will reproduce. So therefore, temperature 37, pH 4 to 4.5, and concentration as high as possible.